Good morning to you all. Today we are going to discuss the fed and fast cycle. The fed, that means we are in a state of feed, has we have taken feed and we are full, and the fast cycle when we are in a state of fasting. And what happens to our metabolism we are going to discuss today. Here you see the mother is ready for with the food and the babies are waiting eagerly. They must have fasted for some time. That is why they are open mouthed and just see the eagerness on their face. What are the different stages of fed and fast cycle? The first one is the well fed state when we are full, then early fasting, then prolonged fasting or starvation, and finally the prolonged starvation. And what are the time period with these different stages of fed and fast cycle? The fed state that lasts for 3 hours after meal ingestion, and the early fasting state, that is post absorptive state, that lasts for from 3 hours to 12 to 18 hours after meal, and the fasting state from 18 hours to 2 days after meal when nothing else is eaten, and the starvation or long term fast that extends to from days to weeks, that is fully adapted to deprivation. Here you see the calories we eat and calories we use. What happens during fed state and what happens during fasted state to our body, our cells, our different tissues, our different organs, and the metabolism in our body of different food materials. In fed state, you see insulin is high, glucose high, burning glucose for energy, and storing fat, which is the extra energy we are that is stored as a fat. And the fasted state, here the insulin is low, glucose is low, liberating fat and burning fat. In fed state we are storing fat, and in fasted state we are burning fat for the energy. Absorptive or well fed state that lasts for from 2 to 4 hours, or averagely you can say 3 hours after normal meal. And what is the other parameters for other things in our body? That is transient increase in plasma glucose, amino acids, and triacylglycerol. And there is increased secretion of insulin and decreased secretion of glucagon. Both are having antagonistic functions. And this absorptive or well fed state is an anabolic state, and all the substrates are readily available. And this elevated insulin or and glucagon ratio causes the increased synthesis of THG, glycogen, and protein, which is stored or utilized our body for structural functions. And all the tissues use glucose as fuel. Metabolic overview, everybody knows carbohydrate, triglyceride, and protein converted to glucose, fatty acids, and amino acids. And again, they are broken down to small carbon chains that enters the Krebs cycle and finally enters the electron transport chain and produces the energy currency, that is ATP. And what is the regulatory mechanism of this fed and fast cycle? There are 4 points, and we have studied during when we were studying discussing enzymes. Number 1 is availability of substrate. Number 2 is allosteric regulation of enzymes. Number 3, covalent modification of enzymes. And number 4 is induction repression of enzyme synthesis through regulation of transcription. Here you see the picture is not very clear, but that shows the time period of all the different mechanism, regulatory mechanism. The availability of substrate, that takes minutes, allosteric modification activators, they take minutes to come into action, and the covalent modification of enzymes that takes minutes to hours, and synthesis of new enzyme molecules, that is induction and repression, it takes hours to days, at least one day. And what is allosteric effectors? After meal, glycolysis is increased because there is plenty of glucose. And this increased fructose 2,6 bisphosphate, which is an allosteric activator of PFK, that is the rate limiting step of glycolysis. What happens when there is glucose is high, and this F2, fructose 2,6 bisphosphate, is the intracellular signal of abundance of glucose, and it is converted from fructose 6 phosphate to fructose 2,6 bisphosphate by the enzyme PFK2. And this F2,6 bisphosphate, when synthesized, and then it acts as an allosteric activator of PFK, and that is the rate limiting step of glycolysis. It activates the PFK and glycolysis is increased. Gluconeogenesis is decreased by F2,6 bisphosphate, which is an allosteric inhibitor of F1,6 bisphosphatase. We know during fed state we are having plenty of substrates, plenty of glucose, so gluconeogenesis is decreased. And but what is the mechanism of this decreased gluconeogenesis? This F1 fructose 1,6 bisphosphate is converted to fructose 6 phosphate and bypassing the PFK1, and the enzyme is this F1 fructose 1,6 bisphosphatase, and this decreased F2,6 bisphosphate is inhibitor of F1,6 bisphosphatase. That is why gluconeogenesis is decreased. Covalent modification, and it is done by phosphorylation and dephosphorylation. Enzymes active in dephosphorylated states are glycogen synthase, PFK2, pyruvate kinase, pyruvate dehydrogenase, and acetyl coenzyme A carboxylase. Glycogen synthase, that is the enzyme for glycogen synthesis. PFK2 we have discussed, this is the enzyme which converts the fructose 6 phosphate to fructose 2,6 bisphosphate. The pyruvate kinase, which is the ninth step enzyme in the glycolysis, pyruvate dehydrogenase, which convert the pyruvate to acetyl coenzyme A in mitochondria, and acetyl coenzyme A carboxylase is the enzyme for fatty acid synthesis. And the exceptions are glycogen phosphorylase kinase and glycogen phosphorylase. Both the enzymes are the enzymes for degradation of glycogen, and the hormone sensitive lipase, which is present in the adipose tissue and causes the lipolysis, the degradation of TAG. Induction and repression of enzyme synthesis. This leads to changes in the number of enzyme molecules, by the transcription. And in well-fed state, what happens? Increased insulin induces the synthesis of key enzymes, that is acetyl coenzyme A carboxylase, that is for fatty acid synthesis, and the fatty acid synthase. And this is induced by the increased insulin. In fast stage, glucagon induces phosphoenol pyruvate carboxykinase of the gluconeogenesis pathway. In that way, the glucagon induces increases gluconeogenesis, and insulin induces the synthesis of fatty acids. And this insulin and glucagon